Okay, let's unpack this. You know how the kidneys are just absolute masters of maintaining balance in our bodies, filtering, well, colossal amounts of fluid every single day.
Right. It's an incredible feat.
It all starts with this really critical first step,
Yeah.
glomerular filtration. And for this deep dive, you specifically asked us to go all the way in, to understand this process, its forces, its regulation, not just surface level, but like the elite professionals who really live and breathe this stuff.
Exactly. Get into the nitty-gritty.
We've got the source material ready, and our mission is to pull out every essential nugget, connect all the dots, and truly grasp this intricate dance happening inside the glomerulus. Ready to jump in?
Absolutely. Glomerular filtration, it's the foundation, isn't it? The very start of urine formation.
Right.
And delving into the why and the how of its precise control, that's absolutely key to a sophisticated understanding of renal physiology.
Yeah.
This isn't just about memorizing numbers, it's about appreciating the elegance of the system design.
So right after this initial filtration step, what's that fluid actually like? Is it basically just blood plasma spilling through?
It's similar to plasma, but, and this is crucial, there are key differences. Glomerular capillaries, unlike many others, are highly impermeable to large molecules, especially proteins.
Okay.
So the fluid that enters Bowman's capsule, we call it the glomerular filtrate, is normally protein-free.
Essentially zero protein.
Pretty much. And it also lacks any cells, no red blood cells, no white blood cells.
Got it.
Now, the concentrations of most small solutes, things like salts, glucose, amino acids, many organic molecules, those are very similar to their concentrations in plasma.
But you hinted there are exceptions, even among the small stuff.
Yes, there are a few important ones. Think about low molecular weight substances that tend to bind to plasma proteins in the blood.
Ah, like calcium.
Exactly like calcium. Almost half the calcium in your plasma is bound to proteins, and most plasma fatty acids are protein-bound too.
So the bound portion can't get through the filter.
Precisely. It's held back with the proteins. So the concentration of total calcium or total fatty acids in the filtrate will actually be lower than in the plasma because only the unbound or free portion is filtered.
Okay, that makes sense. So protein-free, cell-free, and we need to account for the free versus bound state of some smaller molecules. And this filtering happens at an absolutely incredible speed. The sources really emphasize the sheer volume, almost 180 liters a day.
That number, 180 liters, is probably one of the most staggering figures in all of physiology, isn't it?
It really is.
The rate itself, the glomerular filtration rate, or GFR, averages about 125 milliliters per minute in a healthy adult.
125 mil?
Yep. Multiply that out by minutes in a day, and you land right around 180 liters over 24 hours.
But we obviously don't pee out 180 liters.
No, thankfully not. As you know, we typically excrete maybe 1 to 2 liters of urine daily, though that varies a lot with how much fluid you drink.
Right.
Which means that over 99% of that massive initial filtrate volume gets reabsorbed back into the body further down the line in the tubules.
Which just underscores how critical the precision of that initial 180 liters is for all the downstream regulation. Okay, so how does that 125 millimol GFR connect to the total amount of blood actually flowing into the kidneys?
Well, the GFR is actually a fraction of the plasma flow, specifically the plasma part of the blood. It's about 20% of the renal plasma flow.
20%.
We call this the filtration fraction. You calculate it as GFR divided by renal plasma flow.
So 1/5 of the plasma flowing into the kidney gets filtered in one shot.
In a single pass through the glomeruli, yes, about 1/5. And that figure, 1/5, immediately tells you something important.
That the total blood flow must be enormous to supply that much plasma for filtering.
Exactly. It highlights just how extraordinarily high kidney blood flow has to be.
1/5 per pass.
Yeah.
That's really something. Okay, you mentioned earlier the special properties of the glomerular membrane itself that allow this. What makes it so different from, say, a capillary in my arm?
Its structure is just highly specialized for this job of high volume yet selective filtration. It's a 3-layered barrier. Most other capillaries only really have 2 functional layers.
3 layers. Okay, let's walk through them, starting from the blood side and moving outwards.
Okay. First up, you hit the capillary endothelium. That's the inner lining of the capillary. It's fenestrated.
Meaning it's full of little holes.
Yep. Thousands of small pores or fenestrae. Now, you find fenestrations elsewhere, like in the liver capillaries, but these are specifically designed for rapid passage of water and small solutes.
Okay.
And critically, even this first layer, the endothelium, has fixed negative charges on the cell surfaces. These start the process of repelling negatively charged molecules like protein.
Negative charges right off the bat. Okay, layer 2.
Layer 2, surrounding the endothelium, is the basement membrane. Now, this isn't just a simple sheet, it's a complex meshwork. Think of collagen and proteoglycan fibrils woven together.
Like a net.
Sort of, yeah. It has quite large spaces within it, allowing water and small solutes to pass through easily. But it forms a really significant barrier to large proteins, and guess what?
More negative charges.
You got it. It's rich in strong negative electrical charges, mostly from those proteoglycans, so it really reinforces that protein barrier.
Doubling down on repelling proteins. Okay, what's the third and final layer?
The outermost layer is technically the visceral layer of Bowman's capsule, and it's formed by these incredibly specialized cells called podocytes.
Podocytes. Foot cells, right?
Exactly. They aren't a continuous sheet. Instead, they have these long, elaborate, foot-like processes or pedicels that interdigitate. They sort of wrap around the capillaries like fingers clasping a tube.
Okay.
The narrow gaps between these interdigitating foot processes are called filtration slits or slit pores. That's the final hurdle the filtrate has to pass through to get into Bowman's capsule.
And do these podocytes also have negative charges?
They do indeed. The podocytes and their processes also carry negative charges.
So let me recap. A triple-layered barrier. Endothelium with holes and negative charges. Basement membrane meshwork with strong negative charges. And podocytes with these filtration slits between their foot processes, also negatively charged.
That's the structure.
And somehow, despite being thicker than many other capillaries, this barrier filters hundreds of times more fluid. That seems paradoxical.
Well, it does, but that's precisely what highlights its unique design. The overall hydraulic conductivity, basically how easily water moves across it under pressure, is just enormous.
Which brings us to filterability and how both size and charge play a role.
Exactly. Let's unpack that.
Okay, so filterability. If it was just about the size of the molecule, what would we expect?
Well, based purely on molecular size, anything smaller than about, say, 4 nanometers in diameter passes through almost completely freely. Think water, sodium ions, glucose, urea, creatinine. Even slightly larger molecules like inulin, which we sometimes use to measure GFR experimentally, they all have a filterability relative to water of 1.0. They just zip right through.
Okay, and as molecules get bigger,
filterability drops off. For instance, myoglobin, which is a protein but relatively small, around 17,000 molecular weight, its filterability is about 0.75. So 75% as easily filtered as water.
Right. But then you get to albumin, the main plasma protein. Huge molecule, molecular weight around 69,000. Its filterability is practically 0.005, you said?
Vanishingly small, yes.
But its actual diameter, around 6 nanometers, is technically smaller than the estimated size of those filtration slit pores, which are maybe 8 nanometers. So if size were the only factor,
you'd expect way more albumin to leak through than actually does. And this is where the electrical charge becomes absolutely critical.
The negative charges we talked about in the barrier layer.
Precisely. Albumin carries a net negative charge at the normal pH of blood. Those abundant negative charges embedded in the basement membrane and coating the podocytes create a powerful electrostatic repulsion.
It's like trying to push the negative ends of two magnets together.
Exactly that. They actively repel the negatively charged albumin, drastically restricting its passage far beyond what size alone would predict.
The magnet analogy really helps visualize that. And there's experimental proof for this, right? Those dextran studies.
Spot on. Researchers used dextrans, these are inert sugar polymers, and they engineered them to be the same size but carry different electrical charges, positive, neutral, or negative.
Clever.
And the results were clear. Positively charged dextrans filtered much more readily than neutral ones of the same size, and the negatively charged dextrans, they were restricted the most, filtered the least easily.
So that confirms it. The negative charge on the barrier is a crucial functional component, specifically for keeping negatively charged proteins like albumin where they belong in the blood.
Absolutely fundamental.
Okay, so those negative charges are vital. What happens if that charge barrier or the structure supporting it gets damaged? You mentioned minimal change nephropathy earlier.
Yes, minimal change nephropathy is a kidney disease where the glomeruli become leaky to plasma proteins, particularly albumin, even though under a standard light microscope they might look, well, minimally changed or almost normal.
Hence the name.
Exactly. But if you look closer, often with an electron microscope, you see the problem. There's widespread effacement of the podocyte foot processes.
Effacement, meaning they're flattened out?
Flattened, sort of smeared, sometimes detached from the basement membrane. The normal intricate structure of interdigitating foot processes is lost.
Does that physically mess up those filtration slits, widen them or something?
It effectively disrupts the normal architecture of the slit pores, yes. And it's also thought to reduce the effectiveness of the negative charge barrier provided by the podocytes. The exact cause is still debated, but a leading theory involves the immune system, specifically T cells releasing substances, cytokines, that injure the podocytes.
And the direct result of this podocyte injury and effacement is
Increased permeability to proteins. Albumin, being the most abundant plasma protein, starts filtering through in significant amounts and ends up in the urine.
proteinuria or albuminuria.
That's the clinical term, yes. Minimal change disease is most common in young children, but it can happen in adults too, sometimes linked to other conditions. It's just a stark example of how damage to even one part of that complex three-layer filter, in this case, the podocytes and their charge, can severely compromise the whole barrier's integrity.
Okay, so we've got a good handle on the filter itself, its structure, its selectivity based on size and charge. Now let's talk about the actual rate of filtration, that 125 millimin in GFR. How is that rate determined, and more importantly, how is it controlled so precisely?
Right. The GFR itself is determined directly by two main factors. First, the net filtration pressure acting across that glomerular membrane we just discussed. And second, a factor called the glomerular capillary filtration coefficient, which we usually just call KF.
Okay.
The relationship is pretty straightforward, mathematically. GFR equals KF multiplied by the net filtration pressure.
GFR times net pressure. Got it. Let's break down the net filtration pressure first. What are the physical forces involved that pushes and pulls across the filter?
Think of it like the starling forces you see in other capillaries, but the numbers and dynamics are a bit different in the glomerulus. There are four key pressures to consider.
Okay, number one.
Number one, glomerular hydrostatic pressure. Let's call it PG. This is the blood pressure inside the glomerular capillaries. It's the main force pushing fluid out into Bowman's capsule. It favors filtration.
It is quite high.
Yes, estimated around 60 millimeters of mercury in healthy humans. That's significantly higher than in most systemic capillaries.
Oh, PG, pushing out. Number two.
Number two, Bowman's capsule hydrostatic pressure, PB. This is the fluid pressure inside Bowman's capsule, the space receiving the filtrate. This pressure pushes fluid back into the capillaries, so it opposes filtration.
And it's about
Around 18 millimeters of mercury, normally.
All right. PG out, PB in. Number three.
Number three, glomerular capillary colloid osmotic pressure, PHYG. This is the osmotic pressure generated by the plasma proteins, primarily albumin, that are trapped inside the glomerular capillaries. Proteins attract water, so this force pulls fluid back into the capillaries. It opposes filtration.
And its value.
It actually changes along the capillary, but the average value is estimated around 32 millimeters of mercury. We'll come back to why it changes.
Okay, pi G pulling back in. Last one, number four.
Number four, Bowman's capsule colloid osmotic pressure, pi B. This would be the osmotic pressure caused by any proteins in the fluid within Bowman's capsule. Since the filtrate is normally protein-free.
This pressure is essentially zero.
Exactly. So theoretically, it would favor filtration by pulling water out, but in reality, it's negligible, usually considered zero under normal conditions.
Okay, so we have forces favoring filtration, PG, the big one, and B, which is zero, and forces opposing filtration, PB and G.
Correct.
So the net filtration pressure is simply forces favoring minus forces opposing. That works out to PG minus PB minus G.
Precisely. Now let's plug in those typical numbers we mentioned.
Okay, PG is 60 millimeter Hg minus PB, which is 82 millimeter Hg, minus the average G, which is 32 millimeters Hg.
Do the math. 1832 equals 10 millimeters of mercury.
Just 10 millimeters of mercury. That tiny net pressure drives that massive 180 liters per day filtration.
It seems small, doesn't it? But yes, that's the calculated net driving force. And the reason such a small net pressure can produce such a huge volume of filtrate comes back to the other factor in our equation.
KF, the filtration coefficient. It must be incredibly high.
Extraordinarily high. KF essentially represents the permeability and the surface area of the entire glomerular capillary network combined. Think of it as how leaky the filter is per unit of pressure.
How do we measure it?
We can't measure it directly, but we estimate it by rearranging our equation. KFH GFR net filtration pressure.
So 125 millimeter divided by that 10 millimeter Hg net pressure.
Exactly. That gives you a KF of about 12.5 milliliter per minute per millimeter of mercury of filtration pressure for both kidneys combined. Now, to compare it to other tissues, we often normalize it to kidney weight. Per 100 grams of kidney tissue, Kf is about 4.2 milliliter min Hg. And here's the kicker. That value is roughly 400 times higher than the Kf estimated for most other capillary beds in the body.
Wow, 400 times. So that specialized three-layer structure isn't just about selectivity, it's also about creating incredibly high capacity for fluid movement.
That's the key insight. This tremendously high Kf is the major reason the kidneys can filter fluid so rapidly.
So Kf essentially sets the potential for high filtration. Does Kf itself change much physiologically, day to day, as a way to regulate GFR?
Probably not as a primary mechanism for normal daily regulation. Kf is more like a structural property that determines the maximum filtering capacity.
But it can change in disease states.
Yes, definitely. Kf can be reduced in certain kidney diseases. This usually happens in one of two ways, either by reducing the number of functional glomerular capillaries, which decreases the total surface area available for filtration.
Like losing filtering units.
Right. Or by increasing the thickness of the glomerular capillary membrane itself, which reduces its hydraulic conductivity, its permeability.
You mentioned chronic uncontrolled hypertension as an example earlier.
Yes, that's a classic example. Over time, high blood pressure can damage the glomeruli, causing thickening of the basement membrane and eventually leading to the loss of some capillaries entirely. Both processes would reduce the overall capo.
Okay, so Kf sets the stage. It's the high-capacity filter.
Er?
But the net filtration pressure is where the dynamic moment-to-moment regulation happens, mainly by tweaking those forces, Pg, Pb, and B.
That's right.
Let's look at Bowman's capsule pressure, Pb, again. Does that change much under normal physiological conditions?
Normally, no. Pb stays relatively constant around that 18 millimeter Hg mark. While it's true that increasing Pb would oppose filtration and decrease GFR, and decreasing Pb would raise GFR, physiological fluctuations in Pb are not considered a primary way the body fine-tunes GFR.
But pathologically, it can change dramatically with big consequences.
Absolutely. Think about any condition that obstructs the flow of urine after it leaves Bowman's capsule.
Like a kidney stone blocking the ureter.
Exactly. If you get a kidney stone lodged in the ureter, urine backs up. This backup increases pressure all the way back through the tubules into Bowman's capsule, causing Pb to rise markedly.
And if Pb goes up, it directly subtracts from the net filtration pressure.
Correct. It pushes back against Pg. If the obstruction is severe enough, the rise in Pb can significantly reduce or even stop glomerular filtration altogether.
Which can lead to kidney damage, right? Hydronephrosis.
Yes, that pressure buildup can cause the renal pelvis and calyces to swell. That's hydronephrosis, and prolonged obstruction can lead to irreversible kidney damage. It's a very potent clinical example of how a non-regulatory change in just one of these pressures, Pb, can have devastating effects on GFR.
So keeping the outflow path clear is absolutely essential. Okay, let's move to the colloid osmotic pressure in the glomerular capillaries, QB. You said it averages 32 millimeter Hg. Why does it average? Why isn't it constant?
This is where that high filtration fraction, the 20%, really comes into play dynamically. Remember, as blood flows through the glomerular capillaries, from the afferent arteriole towards the efferent arteriole, about 1/5 of the plasma fluid filters out into Bowman's capsule.
But the proteins, like albumin, stay behind in the capillary.
Exactly. Since water leaves but proteins stay, the concentration of proteins in the remaining plasma progressively increases as blood traverses the capillary.
It gets more concentrated as fluid is lost.
Precisely. So if the plasma entering the glomerulus via the afferent arteriole has a colloid osmotic pressure of, say, 28 millimeter Hg,
Okay.
by the time that blood reaches the efferent arterial end of the capillary, having lost 20% of its fluid, the protein concentration is higher, and the colloid osmotic pressure might have risen to around 36 millimeter Hg.
So the average along the whole length is about 32 millimeter Hg.
That's the idea. It's not a single value, but a gradient that increases along the flow path.
And since G opposes filtration, this rising pressure means the force pushing fluid out, Pg minus Pb, is strongest at the beginning of the capillary and gets weaker towards the end as G increases.
That's exactly right. The net filtration pressure actually declines along the length of the glomerular capillary.
Okay, what factors influence this average B?
Two main things determine the glomerular capillary colloid osmotic pressure.
First,
the systemic arterial plasma colloid osmotic pressure itself. If the plasma entering the kidney already has a higher protein concentration for some reason, then the starting G will be higher, the ending G will be higher, and the average BRE will be higher. This tends to decrease GFR.
makes sense. Higher baseline opposition, second factor.
The second and dynamically more important factor for regulation is the filtration fraction. Remember, that's GFR divided by renal plasma flow.
Right, the proportion of plasma being filtered.
If the filtration fraction increases, meaning a larger proportion of the incoming plasma is filtered out, then the proteins in the remaining plasma get concentrated more quickly and more intensely as blood flows through. This causes Ig to rise more steeply and reach a higher final value.
So a higher filtration fraction leads to a higher average D, which in turn opposes filtration and tends to lower GFR. That sounds like a feedback loop involving blood flow.
It is indeed a feedback loop, and this explains a really key concept. Changes in renal blood flow can influence GFR partly through their effect on G, even if glomerular hydrostatic pressure, PG, stays the same.
How so?
Well, imagine renal plasma flow decreases, maybe due to constriction upstream of the glomerulus. If GFR initially tries to stay constant, what happens to the filtration fraction?
GFR stays the same, flow decreases, filtration fraction, GFR flow goes up.
Right. A higher filtration fraction means proteins concentrate faster, G rises more steeply, and this increased opposition from G then acts to reduce GFR.
Ah, I see. So paradoxically, reducing the inflow of blood can actually hinder filtration because you concentrate the remaining plasma too much, increasing the osmotic pull back into the capillary.
That's a major part of the effect, yes.
Yeah.
Conversely, if you increase renal blood flow, the filtration fraction tends to decrease, assuming GFR doesn't jump proportionally. A lower filtration fraction means proteins concentrate more slowly, G rises less steeply, exerting less opposition.
So higher blood flow tends to favor higher GFR, partly because it keeps G lower.
Correct. Even with constant PG, higher flow tends to increase GFR and lower flow tends to decrease it, partly via this effect on paya dynamics.
Wow, that adds another layer of complexity to the whole afferent-tefferent resistance thing we were discussing earlier.
It does. It means effort constriction is particularly complex because it raises PG, but also tends to reduce RBF, which increases filtration fraction and thus raises G. It's a balancing act.
Okay, so we've covered KF, PB, and the dynamic G. Let's finally get to the main driver, the star of the show for physiological regulation. Glomerular hydrostatic pressure, PG. How is that pressure controlled?
You're right, PG, normally around 60 millimeter Hg, is definitely the most important physiologically regulated determinant of GFR. Increases in PG raise GFR, decreases in PG lower GFR. It's quite direct.
And what determines PG itself?
PG is influenced by three main variables, and each of these is under physiological control to some extent.
Variable number one.
Systemic arterial pressure. This is the pressure head driving blood into the kidneys. All else being equal, higher arterial pressure would tend to raise PG and thus GFR.
But you mentioned autoregulation buffers this.
Exactly. As we'll get into more detail soon, the kidneys have remarkable autoregulatory mechanisms that largely shield PG and GFR from normal fluctuations in arterial pressure, but the underlying tendency is there.
Okay. Variable number two, influencing PG.
Efferent arteriolar resistance. This is the resistance in the small artery leading into the glomerulus.
The inflow tap.
Precisely. If you increase resistance here, constrict the afferent arteriole, less pressure gets transmitted downstream into the glomerulus. PG falls and GFR falls.
And if you decrease resistance, dilate the afferent arteriole.
More pressure gets through, PG rises, and GFR rises. Controlling afferent arteriolar resistance is a major point of physiological GFR regulation.
Okay, makes sense. Variable number three.
Efferent arteriolar resistance. This is the resistance in the small artery leaving the glomerulus.
The outflow tap.
Right. Now, think about what happens if you constrict the efferent arteriole, increasing resistance to outflow from the glomerular capillaries.
Blood would back up inside the glomerulus, wouldn't it? Increasing the pressure there.
Exactly. Constricting the efferent arteriole increases resistance to blood leaving the glomerulus, which causes pressure within the glomerular capillaries, PG, to rise.
So efferent constriction increases PG, which should increase GFR.
It does initially, yes. Moderate increases in efferent resistance tend to increase GFR slightly because the rise in PG outweighs other effects.
But you use cautious words there. Initially, slightly, moderate. What happens if the efferent constriction is severe?
This is where that biphasic effect we hinted at comes in, and it's crucial for the deep understanding you're aiming for. Yes, severe efferent constriction, say, increasing resistance more than threefold does raise PG even further. However, It also dramatically reduces the total renal blood flow passing through the glomerulus.
Okay, flow goes way down.
And when flow goes way down, but filtration continues, driven by that high PG, what happens to the filtration fraction?
It skyrockets. You're filtering a much larger percentage of the very small amount of plasma getting through.
Exactly. And what happens when the filtration fraction skyrockets?
The protein concentration in the remaining capillary plasma goes up incredibly fast, meaning PG rises very, very steeply along the capillary.
Bingo. With severe efferent constriction, the rise in colloid osmotic pressure becomes so extreme that it actually overwhelms the increase in hydrostatic pressure, PG, caused by the constriction itself.
So the force pulling fluid back in becomes stronger than the increased force pushing it out, PG.
Precisely. The net filtration pressure actually decreases, and GFR paradoxically falls below normal, sometimes significantly.
Wow.
The main culprit is that amplified nonlinear rise in TG, partly due to the Donan effect adding extra osmotic pull from ions bound to the concentrated proteins. So, efferent constriction is a double-edged sword. Moderate constriction slightly boosts GFR, but severe constriction tanks it because of the overwhelming rise in PG due to reduced flow.
That is a fascinatingly complex interplay between pressure and flow dynamics. Okay, so afferent constriction always drops GFR. Efferent constriction has this biphasic effect depending on severity, impacting both PG and TG. You got it. Let's shift focus slightly to the overall renal blood flow, RBF itself. You mentioned it has to be huge to support that GFR. How huge are we talking?
We're talking about 1,100 milliliters per minute, combined for both kidneys, in an average 70-kilogram person.
Over a liter of blood every minute.
Yes, and think about this. That's roughly 22% of the entire cardiac output.
22%. For organs that make up, what, less than 0.5% of body weight?
Exactly. The kidneys weigh only about 0.4% of total body weight, yet they receive almost a quarter of the blood pumped by the heart. It's an absolutely enormous blood supply compared to almost any other organ on a per-weight basis.
Why do they need so much? Is it just to meet their own energy needs?
No, not at all. The amount of blood flow far, far exceeds what they need just for delivering oxygen and nutrients for their own metabolism, unlike, say, muscle or brain tissue.
So what's the primary purpose of all that extra flow?
It boils down to one thing: supplying enough plasma to support that incredibly high rate of glomerular filtration. We keep talking about the 125 milliburnum and 180 LD.
The flow is there to feed the filter.
Precisely. That constant high-volume filtration is the cornerstone of the kidney's main job, precisely regulating body fluid volumes and the concentrations of various solutes. To do that effectively, they need to filter a lot first, then selectively reabsorb or secrete. So the mechanisms controlling renal blood flow are, as you expect, intimately linked to controlling GFR and overall kidney excretory function.
That high flow also explains something else you mentioned, the oxygen consumption. Kidneys consume a lot of oxygen per gram of tissue, but because the blood flow is so massive, the difference in oxygen content between the renal artery and renal vein is actually quite small, right?
That's exactly right. They extract a relatively small percentage of the oxygen delivered, but because the delivery rate, flow times arterial oxygen content, is so huge, the total amount of oxygen consumed is still very high.
What are they using all that oxygen for, if not just basic cell maintenance?
A very large fraction, the majority, in fact, of the oxygen consumed by the kidneys is directly tied to the high rate of active sodium reabsorption happening in the renal tubules, downstream from the glomerulus.
Pumping sodium back into the blood after it's been filtered.
Yes. Remember, huge amounts of sodium are filtered. The kidney cells lining the tubules have to actively pump most of that sodium out of the filtrate and back into the blood. That active transport requires a lot of energy, ATP, which requires oxygen to produce.
So there's a direct link. GFR determines how much sodium gets filtered. Tubular reabsorption then has to pump most of that filtered sodium back, and the energy cost of that pumping dictates oxygen consumption.
You've nailed it. Renal oxygen consumption varies almost in direct proportion to the rate of tubular sodium reabsorption, which itself is tightly coupled to GFR and the filtered sodium load.
What happens if GFR drops to 0?
If glomerular filtration completely ceases, then there's no sodium being delivered to the tubules to be reabsorbed. Renal sodium reabsorption stops, and oxygen consumption plummets down to a basal level maybe only about 1/4 of normal. That basal level just covers the basic metabolic needs of the kidney cells themselves to stay alive.
That really underscores how tightly filtration, reabsorption, and the kidney's energy budget are interwoven.
It's a beautifully integrated system.
Okay, so the massive blood flow is there to supply plasma for filtration, and the kidney's energy use is largely driven by reabsorbing what gets filtered. How is the total renal blood flow, RBF, itself determined fundamentally?
Fundamentally, it's just like blood flow in any other tissue. RBF is determined by the pressure gradient across the renal vasculature divided by the total resistance within that vasculature.
Ohm's law for fluids, flow, pressure resistance.
Exactly. So it's renal artery pressure minus renal vein pressure divided by the total renal vascular resistance.
And renal artery pressure is basically systemic arterial pressure.
Pretty much, yes. And renal vein pressure is quite low, typically only around 3 to 4 millimeters of mercury. So the main pressure gradient is driven by systemic arterial pressure.
Okay. And where does most of the resistance reside within the kidney's blood vessels? Where are the control points?
While there's some resistance in all the vessels, the vast majority of the total renal vascular resistance is located in 3 major segments: the interlobular arteries, and then most importantly for regulation, the afferent arterioles and the efferent arterioles.
The inflow and outflow taps again.
Those are the key adjustable resistance points. The resistance in these vessels is under complex control by the sympathetic nervous system, various hormones, locally produced substances called autocoids, and, as we've touched on, intrinsic kidney mechanisms. Increasing resistance in any of these segments reduces RBF, while decreasing resistance increases RBF, assuming arterial and venous pressures stay constant.
And you keep mentioning that despite changes in arterial pressure, RBF and GFR tend to stay remarkably constant over a certain range. That's autoregulation, right?
Correct. The kidneys possess incredibly effective mechanisms to maintain both renal blood flow and GFR relatively constant, despite significant fluctuations in arterial blood pressure, typically within a range of about 80 up to 170 or 180 millimeters of mercury.
And this autoregulation is intrinsic. It happens within the kidney itself.
Yes, that's the amazing part. These are feedback mechanisms that operate within the kidney tissue. They function even in kidneys that have been removed from the body and perfused with blood, demonstrating they are independent of systemic nerves or circulating hormones, although those systemic factors can certainly modulate or even override autoregulation in specific situations.
That's fascinating. And unlike autoregulation in, say, muscle, which is focused on matching blood flow to metabolic demand, oxygen, nutrients.
Right.
The kidney's autoregulation has a different primary purpose, doesn't it? Because its flow is already way above metabolic needs.
Exactly. The major function, the primary goal of renal autoregulation is to maintain a relatively constant GFR.
Why is keeping GFR stable so important?
Because it allows for precise control of the renal excretion of water and solutes downstream in the tubules. Imagine trying to fine-tune reabsorption and secretion if the amount being filtered was constantly swinging wildly up and down with every little change in blood pressure. It would be chaos.
You'd overshoot or undershoot constantly.
Precisely. A stable GFR provides a stable starting point, a predictable filtered load, upon which the tubules can then exert their fine regulatory control to match excretion with intake and body needs.
Just how effective is this GFR autoregulation?
It's remarkably effective. GFR normally stays relatively constant, changing by less than 10%, even when mean arterial pressure fluctuates quite widely, say from as low as 70 or 75 millimeter Hg up to 160 or even 180 millimeter Hg.
That's a huge pressure range with minimal GFR change.
It is. Renal blood flow is generally autoregulated in parallel with GFR over this range, although under some conditions, GFR autoregulation might be slightly more efficient or operate over a slightly wider range than RBF autoregulation.
Can you remind us of that striking example from the source material about what would happen without it? The urine output calculation?
Ah, yes, the quantitative argument. It really drives home the importance. So normal GFR is 180 L-day. Normal tubular reabsorption is about 178.5 L-day, leaving 1.5 L-day of urine output. Now, imagine there was no autoregulation. A relatively modest increase in arterial pressure, say from 100 to 125 millimeter Hg, a 25% increase, might cause a proportional 25% increase in GFR, from 180 up to 225 L-day.
Right.
If tubular reabsorption somehow magically stayed fixed at 178.5 L-day, what would urine flow become?
225 minus 178.5, that's 46.5 liters per day.
Exactly. A more than 30-fold increase in urine output from just a 25 millimeter Hg rise in blood pressure?
You deplete your entire plasma volume, which is only about 3 liters, in a matter of hours.
It would be catastrophic, yes. Thankfully, reality is very different. Changes in arterial pressure have much, much smaller effects on urine volume.
Two main reasons, right?
Two main reasons. First, as we've been discussing, renal autoregulation does exist, and it effectively prevents those massive GFR swings in response to pressure changes.
It buffers the GFR.
Yes. And second, there are additional adaptive mechanisms in the renal tubules themselves. One key one is called glomerular tubular balance, where the tubules tend to increase their rate of reabsorption when GFR increases.
So reabsorption tries to keep pace with filtration to some extent.
It does. But it's important to note, even with both autoregulation and glomerular tubular balance working, changes in arterial pressure do still have significant effects on the renal excretion of water and sodium, especially over the longer term.
And that's pressure diuresis and pressure natriuresis.
Precisely. This residual effect of pressure on excretion is absolutely fundamental to the long-term regulation of body fluid volumes and, indeed, the long-term regulation of arterial pressure itself. But the acute, potentially disastrous swings are prevented by autoregulation.
Okay, so autoregulation is key for providing that stable GFR baseline. What are the specific intrinsic mechanisms within the kidney that are responsible for this autoregulation?
The most important and best understood intrinsic mechanism is called the tubuloglomerular feedback mechanism, or TGF.
Tubuloglomerular feedback, the tubule talking back to the glomerulus.
That's a great way to put it. It's a very special feedback system unique to the kidney that links changes in the composition of the fluid flowing through the distal part of the tubule, specifically the sodium chloride concentration, with the control of resistance in the renal arterioles, both afferent and efferent, that feed the glomerulus.
And its main goal isn't just GFR stability per se.
Its primary goal seems to be ensuring a relatively constant delivery of sodium chloride to the distal tubule. The distal tubule is where the final, fine-tuned adjustments to urine composition happen. TGF helps prevent spurious fluctuations in salt delivery to this critical site, allowing it to do its job properly.
So how does this talking back work? What's the anatomy involved?
It relies on a specialized structure called the juxtaglomerular complex, or juxtaglomerular apparatus, JGA.
Juxtaglomerular meaning next to the glomerulus.
Exactly. The JGA consists primarily of two cell types. First, the macula densa cells. These are a plaque of specialized epithelial cells located in the very first part of the distal tubule, right where it passes between the afferent and efferent arterioles of its own glomerulus.
Ah, so the tubule loops back and snuggles right up against its own blood supply origin.
Perfectly positioned. Their structure strongly suggests they are sensing the tubular fluid composition and secreting signaling molecules towards the arterioles.
Okay, macula densa cells sensing the fluid. What's the other cell type in the JGA?
The other key players are the juxtaglomerular cells, or JG cells. These are specialized, smooth muscle cells located primarily in the walls of the afferent arteriole and, to some extent, the efferent. Crucially, these JG cells are the main site where the enzyme renin is synthesized, stored, and released.
Renin, the start of the renin-angiotensin system.
The very same. Okay, so you have the macula densa sensing NaCl in the tubule right next to the JG cells in the arterioles, which hold renin.
How does the feedback loop operate? Let's say GFR decreases.
Okay, if GFR decreases, for whatever reason, maybe arterial pressure drops slightly, the flow rate of filtrate through the nephron, including the loop of Henle, slows down.
Flow.
This slower flow gives the thick ascending limb of the loop of Henle more time to actively reabsorb sodium and chloride ions out of the tubular fluid before that fluid reaches the macula densa.
So more NaCl gets pulled out before it gets to the sensor.
Exactly. Which means the concentration of sodium chloride in the fluid arriving at the macula densa cells will be reduced.
Okay, so the macula densa senses low NaCl. What signal does that trigger?
This decrease in macula densa NaCl concentration initiates a signal from the macula densa cells that has two key effects on the adjacent arterioles.
Effect number one.
Effect number one. It causes a decrease in the resistance of the afferent arteriole. It signals the afferent arteriole to dilate.
Afferent dilation increases inflow pressure, PG, right?
Correct. It helps restore glomerular hydrostatic pressure and thus brings GFR back up towards normal.
Okay. And effect number two from low NaCl signal.
Effect number two, it stimulates the juxtaglomerular JG cells in the arteriolar walls to release renin.
And renin kicks off the angiotensin cascade.
Right. Renin leads to the formation of angiotensin I, which is quickly converted to angiotensin II. And what does angiotensin II do to the efferent arteriole?
It constricts it. Preferentially constricts the efferent arteriole.
Exactly. And efferent arteriolar constriction also tends to increase glomerular hydrostatic pressure, PG.
Ah, so both components work together. Low NaCl at the macula densa leads to afferent dilation and renin release, causing efferent constriction via ANG2. Both actions raise PG to counteract the initial drop in GFR.
Precisely. This dual-component tubuloglomerular feedback mechanism operating via the JGA is incredibly elegant and effective at stabilizing GFR when challenged by changes in perfusion pressure.
That is a beautiful feedback system. Is TGF the only intrinsic mechanism responsible for autoregulation, though?
No, there's another mechanism believed to contribute, especially to the RBF aspect, and perhaps more to protecting against sudden pressure surges. It's called the myogenic mechanism, or myogenic response.
Myogenic, muscle originating.
Yes. It's an intrinsic property of vascular smooth muscle itself, particularly in small arterioles like the afferent arteriole. These muscle cells have the ability to contract when they are stretched.
So if arterial pressure goes up, the wall of the afferent arteriole gets stretched.
And the smooth muscle cells in that wall respond directly to that stretch by contracting.
How does the stretch trigger contraction?
The stretch is thought to open stretch-activated calcium channels in the smooth muscle cell membrane. Calcium flows into the cell, triggering the contractile machinery.
So pressure rises, arterial stretches, muscle contracts, vessel constricts, resistance increases.
That's the sequence. This increased resistance counteracts the initial rise in pressure, helping to prevent that pressure from being fully transmitted downstream to the delicate glomerular capillaries.
So it helps buffer the pressure hit on the glomerulus.
Exactly. It helps prevent excessive increases in both renal blood flow and GFR when arterial pressure suddenly rises. While its exact contribution to the steady-state autoregulation compared to TGF is sometimes debated, the myogenic mechanism is likely very important for providing a rapid, almost instantaneous defense against hypertensive damage to the glomeruli. It acts within seconds.
Okay, so we have TGF as the fine-tuning NaCl sensing regulator, and the myogenic response as a more direct, rapid physical buffer against pressure changes, particularly sudden increases.
That's a good way to summarize their likely complementary roles, yes. Both are intrinsic to the kidney.
We've established how these sophisticated intrinsic mechanisms work hard to keep RBF and GFR stable, despite blood pressure fluctuations. But are there other conditions or factors beyond just blood pressure that can significantly alter RBF and GFR, perhaps by interacting with these various systems?
Oh, yes, definitely. While autoregulation provides stability against pressure changes, other physiological or pathological states can indeed influence these rates, often by modulating the TGF system itself.
Like diet. You mentioned high protein intake earlier.
Yes, that's a classic example. It's well known that a high protein intake increases both renal blood flow and GFR. Now, part of this effect, if it's chronic, is due to actual growth of the kidneys. But even acutely, within just an hour or two after eating a high protein meal, GFR and RBF can increase by 20 to 30%.
Why would eating steak make your kidneys filter faster?
The most plausible explanation ties directly back to that tubuloglomerular feedback mechanism we just discussed.
How so?
A high-protein meal leads to increased absorption of amino acids from the gut, raising amino acid levels in the blood. These amino acids are then filtered at the glomerulus and subsequently reabsorbed in the proximal tubules.
Okay.
Now, many amino acids are reabsorbed via co-transport mechanisms that are coupled with sodium reabsorption. So when there's a high load of amino acids to reabsorb, this process also drives an increase in sodium reabsorption in the proximal tubule.
So more sodium gets pulled back early in the proximal tubule.
Which means less sodium chloride is left in the tubular fluid to travel downstream and reach the macula densa.
Less NaCl at the macula densa. That's the same signal as a drop in GFR.
Exactly. The decreased NaCl concentration arriving at the macula densa triggers the standard TGF response. Decreased resistance in the afferent arterioles dilation.
Which raises RBF and GFR.
Precisely. This increase in GFR serves a couple of purposes. It helps to maintain overall sodium excretion near normal levels, despite the increased proximal reabsorption. And importantly, it increases the filtration and subsequent excretion of the waste products of protein metabolism, like urea, which primarily rely on GFR for elimination.
That's a fascinating link. Diet directly manipulated the GFR set point via the TGF mechanism. Does a similar logic explain why GFR is often elevated in people with uncontrolled diabetes mellitus?
It's believed to be a very similar mechanism, yes. In uncontrolled diabetes, you have high blood glucose levels. Glucose is also filtered freely at the glomerulus.
And reabsorbed in the proximal tubule along with sodium, right? Via SGLT cotransporters.
Correct. When there's a very high filtered load of glucose, the proximal tubules work overtime to reabsorb it, and this process drags extra sodium along with it, increasing proximal sodium reabsorption.
Just like with the amino acids. Increased proximal sodium uptake means less NaCl delivered to the macula densa downstream.
Exactly the same consequence. The macula densa senses low NaCl, triggers afferent arteriolar dilation via TGF, and RBF and GFR increase, often quite significantly in poorly controlled diabetes.
So both high protein intake and hyperglycemia seem to hijack the same feedback loop, pushing GFR higher by tricking the macula densa into thinking filtration is too low.
That appears to be the common pathway. And these examples really highlight something fundamental about TGF. Its main purpose might not be to keep GFR absolutely constant under all circumstances, but rather to work towards ensuring a relatively constant delivery of sodium chloride to the distal tubule.
Why is constant distal NaCl delivery so important?
Because the distal tubule and collecting duct are where the final, highly regulated adjustments of sodium, potassium, water, and acid-base balance occur, they need a relatively stable inflow of solute to function optimally and make the precise adjustments needed to maintain overall body homeostasis. TGF helps provide that stability.
What about the flip side? What if proximal tubule reabsorption is decreased, maybe due to damage?
Then you see the opposite sequence, again mediated by TGF. If the proximal tubules are injured, say by toxins like heavy metals or certain drugs, and can't reabsorb sodium chloride properly,
then large amounts of NaCl would flow past them and reach the distal tubule and macula densa.
right, without any compensation, this would lead to massive salt and water loss, quickly causing volume depletion.
So what does TGF do in response to high NaCl at the macula densa?
It signals the afferent arterial to constrict. This renal vasoconstriction reduces RBF and GFR.
Lowering the filtered load to compensate for the poor proximal reabsorption.
Exactly. It's another compensatory response mediated by TGF, again emphasizing its role in trying to normalize solute delivery to the distal nephron segments, protecting the body from drastic fluid and electrolyte imbalances when other parts of the system are compromised.
It's really a sophisticated regulator. Beyond these specific dietary or disease examples, are there other sort of broader factors that generally influence RBF and GFR levels, like age?
Yes, age is definitely a factor. GFR and RBF are actually lower in newborns and infants, gradually increasing to reach typical adult levels by around the age of 2 years.
And then later in life.
After about the fourth decade, so around age 40, both GFR and RBF tend to gradually decline. A rough estimate is a decrease of about 5% to 10% per decade thereafter, although there's significant individual variability.
What causes that age-related decline?
It's likely multifactorial. It coincides with a gradual loss of functional nephrons over time, perhaps related to cumulative low-grade injury from factors like increasing blood pressure, metabolic changes, increased oxidative stress, maybe some age-related decline in vasodilators like nitric oxide.
Are there known differences between sexes?
Yes, on average, adult men tend to have slightly higher RBF and GFR than women, even after correcting for differences in body size.
Interesting.
However, men also tend to experience a somewhat more rapid decline in GFR as they age compared to premenopausal women.
Any idea why?
Hormones? The exact reasons aren't fully clear, but hormonal influences are often suggested. For example, estrogens might have some protective effects on the kidneys, while androgens might have some detrimental effects over the long term, potentially contributing to these observed sex differences in GFR decline.
Are there other conditions that can influence RBF and GFR?
Sure. Things like obesity, chronically high salt intake, administration of glucocorticoid hormones, even just having a fever can potentially influence renal hemodynamics. Though for some of these, like obesity or high salt intake, the long-term effects might involve actual glomerular injury and pathology rather than just functional changes.
We should probably also loop back quickly to the systemic signals, the nerves and hormones. Even though autoregulation tries to buffer them, how do they fit in?
Right. While autoregulation handles the intrinsic stability, systemic factors absolutely play a role, especially when the body is under stress or needs to make major adjustments.
Like the sympathetic nervous system.
Yes. Almost all the blood vessels in the kidney, including those critical afferent and efferent arterioles, receive input from sympathetic nerve fibers.
And strong activation, like fight or flight.
Strong sympathetic activation, think severe hemorrhage, major stress, brain ischemia, causes significant constriction of both afferent and efferent arterioles via norepinephrine release. This markedly reduces both RBF and GFR.
Why reduce GFR in an emergency?
To conserve body fluid volume and help maintain systemic blood pressure when it's threatened. It's a way of prioritizing circulation to vital organs like the brain and heart over kidney filtration.
What about milder sympathetic activity? specifically? Does it do much to RBF-GFR?
Interestingly, moderate or mild sympathetic stimulation seems to have relatively little effect on overall RBF or GFR. Normal reflex adjustments, like from baroreceptors sensing small pressure changes, don't typically cause big shifts in kidney hemodynamics.
But mild sympathetic activity can still affect the kidney.
Yes, it can stimulate renin release from the JG cells, and it can also directly increase sodium and water reabsorption by the tubules, thus reducing urinary excretion even without changing GFR much.
So strong sympathetic hits RBF-GFR hard for emergencies, while mild activity might focus more on saltwater balance and renin.
That seems to be the general pattern, yes. The nerves are most important for overriding autoregulation during severe acute disturbances.
What about key hormones? We keep coming back to angiotensin II. Let's solidify its role.
Angiotensin II is arguably the most powerful and physiologically significant hormonal regulator of renal hemodynamics, particularly in situations that challenge blood pressure or volume.
Like low blood pressure and dehydration.
Exactly. Ang2 acts as both a circulating hormone and can also be produced locally within the kidney as an autocoid. Receptors are widespread, but as we discussed, its functional effect is most pronounced on the efferent arterioles.
Why the preference for efferents again?
It seems the afferent arterioles are somewhat protected from Ang2's constrictor effect under many physiological conditions, possibly due to simultaneous release of local vasodilators like nitric oxide or prostaglandins that counteract it there. The afferent arterioles lack this protection or are just more sensitive.
So Ang2 preferentially constricts the outflow tab.
Yes, and by doing so, it helps to raise PG, the glomerular hydrostatic pressure.
Which helps maintain GFR even if overall RBF is reduced due to the efferent constriction.
Precisely. This is its crucial adaptive role. When arterial pressure drops or volume is depleted, conditions that would normally decrease. GFR increased Ang2 levels kick in, constrict the efferents, prop up PG, and thus help prevent a catastrophic fall in GFR.
Allowing the kidney to keep filtering waste products like urea and creatinine.
Exactly. While at the same time, the reduced overall RBF caused by the efferent constriction leads to slower flow in the peritubular capillaries downstream, which actually enhances the reabsorption of sodium and water from the tubules.
So it's a brilliant dual effect. Maintain waste filtration and promote salt and water retention to help restore blood volume and pressure.
It's a cornerstone of how the kidney integrates pressure regulation, volume regulation, and filtration. It's also involved in modulating the sensitivity of the TGF response itself.
And this also explains why drugs that block angiotensin II, like ACE inhibitors or ARBs, can sometimes cause a sharp drop in GFR, especially in patients who rely on that Ang2 efferent constriction to maintain their kidney function.
Absolutely critical clinical point. In patients with conditions like bilateral renal artery stenosis, where kidney perfusion pressure is already low, they become highly dependent on Ang2, constricting the efferent arterioles to maintain GFR. Blocking Ang2 in these patients can remove that support, leading to a precipitous drop in GFR and potentially acute renal failure. It highlights why monitoring kidney function is so vital when starting these otherwise very beneficial drugs.
A direct link from physiology to clinical practice. What about hormones or autocoids that do the opposite, cause vasodilation?
Yes, there are important vasodilators too, which decrease renal vascular resistance and tend to increase both RBF and GFR.
Like nitric oxide.
Nitric oxide, or NO, is a key one. It's produced by the endothelial cells lining the blood vessels throughout the body, including the kidneys. There's a basal tonic level of NO production that's essential for maintaining normal low vascular resistance in the kidneys, keeping them vasodilated.
What happens if NO production is blocked?
If you inhibit NO synthesis, renal vascular resistance increases, RBF and GFR decrease, sodium excretion falls, and this can actually contribute to developing high blood pressure. It's thought that impaired NO production might play a role in the increased renal vasoconstriction seen in conditions like chronic hypertension or atherosclerosis.
Okay, so NO keeps things relaxed. Any others?
Prostaglandins, specifically PGE2 and PGI2 and another substance called bradykinin, also act as vasodilators in the kidney, increasing RBF and tending to increase GFR.
Are they major day-to-day regulators?
Perhaps not under normal resting conditions, but they become very important because they can act as local buffers or counter-regulators. They specifically seem to dampen or oppose the vasoconstrictor effects of the sympathetic nerves and angiotensin II, particularly on the afferent arterioles.
So they protect the inflow vessel from clamping down too hard.
Exactly. By opposing excessive afferent constriction during stressful situations like volume depletion, surgery, etc., Prostaglandins help prevent potentially damaging reductions in GFR and renal blood flow.
Which explains why taking NSAIDs, non-steroidal anti-inflammatory drugs like aspirin or ibuprofen, which inhibit prostaglandin synthesis, can sometimes cause kidney problems.
Precisely. Under normal circumstances, blocking prostaglandins might not have much effect. But in situations where the kidneys are stressed and relying on those prostaglandins to counteract vasoconstrictor influences, taking an NSAID can remove that protective buffer, leading to unopposed afferent constriction and potentially a significant drop in GFR.
Wow. Okay, we have truly done a deep dive here. We've gone from the incredibly intricate three-layered filter itself, understanding its size and charge selectivity, to the complex balance of starling forces. The hydrostatic pressures Pg and Pb, the crucial colloid osmotic pressure G, and how it changes dynamically with filtration fraction.
Right. The dynamic nature of Pg is key.
We've appreciated the immense filtering capacity represented by that huge K-off value, and then we explored the layers of control. The primary regulation of Pg via afferent and efferent arterial resistance.
The inflow and outflow tap.
The genius of the tubuloglomerular feedback system sensing distal NaCl delivery via the macula densa and JGA, the protective myogenic response buffering pressure surges,
Intrinsic mechanisms.
and then how systemic signals like sympathetic nerves and hormones, especially angiotensin II, integrate with these local controls, and even how things like diet and diseases like diabetes can tap into and influence these very same regulatory pathways.
It truly is a system of remarkable precision, integration, and adaptability. To really get that elite professional grasp of renal physiology, mastering these interlinked determinants and regulatory mechanisms of glomerular filtration is absolutely fundamental. You've really dug into the core mechanics.
And by breaking it down, layer by layer, the structure, the forces, the coefficients, the feedback signals, the differential effects on afferent versus efferent vessels, the role of the macula densa, you start to see how it all works together, not just that it works.
Absolutely. The details build the complete functional picture.
So left pull back for a final thought. What does all this mean for kidney health in the bigger picture? We've seen how vital it is to maintain a stable GFR and how powerfully the kidney defends that GFR using autoregulation and hormones like ANG2, especially when pressure is low.
Right, those are protective mechanisms.
But we also saw how other factors, like uncontrolled high blood sugar or maybe even chronically high protein intake, can persistently increase GFR by manipulating the TGF signal, essentially telling the kidney to filter more all the time.
Yes, leading to chronic glomerular hyperfiltration.
So here's the provocative question to leave you with. Considering how finely tuned autoregulation normally is to protect the glomerulus from excessive pressure, and yet how these metabolic signals can override it and drive GFR chronically high, how might that sustained hyperfiltration itself, that constant state of elevated glomerular pressure and flow driven by signals like hyperglycemia, eventually contribute to the physical damage, the scarring, the glomerulosclerosis that we see in the progression of chronic kidney disease associated with conditions like diabetes or obesity?
It's a critical question. Could the very feedback systems designed for short-term adaptation become maladaptive and contribute to long-term injury when persistently activated outside their intended physiological range? It really challenges us to think about the potential double-edged sword of these powerful regulatory mechanisms when we consider lifelong kidney health.